Hi, this is Dr. Dan Guerra coming to you from Diaeventontology and Authentic Biochemistry Studios in the beautiful inland Pacific Northwest, which is just outside my window behind me, but you can't see much because it is very cloudy and rainy and it's going to turn to snow. So the backdrop isn't nearly as beautiful as it might be, although if I put the camera out there, I think you would see that it's still very nice. So let's get started here. What I want to do is bring forward a lot of the work I've been discussing in my Authentic Biochemistry podcast, which is a series of lectures where we've been discussing autoimmune diseases, particularly talking about how they are mediated by ontological phenomena that include things like inflammation to a specific antigen that is a host antigen that can be triggered by multiple methods, but all of them either endo or exo-environmental. By endo, I mean internal systems becoming dysregulated, or external systems, that is the inculcation or the introduction of, say, a parasite or a pathogen or some other kind of stress phenomena. That then can lead to an overreaction of the immune system. And if it's occurring in any aspect of regulation of metabolism or of cellular signaling or indeed of simple gene expression in specific cell types that can be associated with the central nervous system, either peripherally and then signaling to the CNS or directly within the CNS, as with microglia or neurons, then you can then induce the potential for a neuroimmune inflammatory response, which then can trigger an epigenetic phenomena, which ultimately then can cause neurodegeneration, for example, such as in demyelinating uh, diseases like MS, or neurodegeneration associated with demyelination, like with Alzheimer's and Parkinson's disease, or indeed also with Graves' disease. Several diseases that we're going to discuss again as we go through these video lectures. So, anyway, that is a very brief prolegomena for a very long series of lectures I'm going to be doing now. Again, I'm Dr. Daniel John Guerra, and this is VeraMed Authentic Biochemistry Studios. The date is the 7th of January, and a brand new year, the 2020. So we're now starting the third decade of the 21st century. Kind of amazing. At any rate, those are the various websites and links you can find, either the YouTube channel where I'm posting these free lectures, and also the two other places where you can find my podcasts, and again, my Facebook page down here, where I'm posting all of my authentic biochemistry and combined VeraMed video lectures podcasts so that you can get those for free. Obviously, I don't work for free. I have the ability to be hired by professionals and by lay people. And what you hire me for is to delve deeply and to verify evidence within the published scientific research, usually associated with biochemistry, cell physiology, general physiology, human physiology, genetics, molecular genetics, epigenetics, under the umbrella, sometimes I call it just simply biomedicine, because it is going to be human based. The literature I've been looking at for my clients have been primarily about human based, although we have looked at microbial genetics and molecular genetics, as well as plant biology for some of my clients. So, again, today we're going to look at Diaeventontology. That is a new paradigm I've created for an understanding of scientific phenomena. And I've talked about it at great length in various other lectures formats. We'll bring it up again here, maybe not today, but basically, if you tear apart the term, what I want to look at is the dialectics of events, and those events within an ontological perspective, that is, looking at the metaphysics of how phenomena can come together and form what we understand as scientific phenomena, and then our description of that scientific phenomena utilizing this new paradigm of looking at aspects of the environment, the neuroimmune and neuroepiimmune phenomena within environmental changes induced by environmental changes, And then with the backdrop of standard genetics, that is inheritance of genes, and then overall the changes in gene expression and how they can be modified in disease states, and a better understanding of that through time, which is where the event aspect comes in, denominator being time, rather than simply substance ontology. We can hopefully get a better handle on describing both healthy biological phenomenon in the backdrop of human physiology and human health, as well as when things go awry, when we get dysfunction and disease. So that's a very brief overview of what I mean by Diaeventontological perspectives in biomedicine. So, at any rate, without any further discussion of that, let's just get going. All right. So, we've been discussing this particular paper, this 2019 paper, in my Authentic Biochemistry podcast. And we're just going to go back and take a look at it again. This is the International Journal of Molecular Science 2019. And let's take a look at what some of the aspects of the focus of this paper are. Okay? So, sphingolipids are membrane and signaling acyl lipids. Acyl lipids meaning they are associated with an acyl group, which is a carbonyl oxygen bonded to another molecule so that you have an acyl radical. And that's usually a fatty acid, okay, in biology. So you have acyl lipids as opposed to prenolipids, which are all part of the huge family of prenolipids, sterols, such as cholesterol, as being one of the parent molecules, at least in humans. So we're talking about acyl lipids here today. And the primary roles in cell fate and neurotransmission. There are two key metabolically active components of that complex pathway. And it is a very complex pathway, but don't worry, we're going to be able to disambiguate it. And those two important lipids we want to talk about are ceramide versus sphingosine. Now, they're very closely related structurally, but they have quite divergent biological roles. Not necessarily contradictory, but contrary roles in biological aspects. They have different receptors, for example. Those receptors will mediate things downstream, such as signal transduction cascades, phosphorylation, ubiquitinylation, acetylation, methylation at the DNA level, histone level, and then altering gene expression ultimately, either transcriptionally or post-translationally by altering protein synthesis, degradation, movement, trafficking through the cell or out of the cell or back in the cell. So you have ceramide and sphingolipids, and they're the backbone structures of all the sphingolipids, basically. And I'll show you the structures, don't worry, because I am a biochemist. In fact, I'm a lipid biochemist. I love lipid structures, right? So, brief overview right now. Ceramide participates in a wide variety of biochemical events, such as protein phosphorylation, modulation of protein kinase C, really important enzyme which adds phosphate to other proteins and thus inducing a signal transduction cascade intracellularly after some signaling, usually at the level of membrane-associated ligand binding to a receptor. And also phospholipase A2 is being mediated by this response. And phospholipase A2 just means that you're removing an acyl fatty acid from the 2 position normally of a glycerol phospholipid, such as phosphatidylcholine, phosphotidylserine, phosphoethynolamine, phosphotidylinositol, associated in the membrane, of the plasma membrane, that is. But besides that, ceramide also mediates signal transduction, leading to cell death. So ceramide is a very important cell death signaling compound, lipid, okay? And in high concentration, that's while sphingolipids and cytosine, which I'll show you the structure in a minute and give you much more detail, are potent actually inhibitors of protein kinase. So I told you that they're going to be contrary, not necessarily contradictory in terms of biological, biochemical motifs, signaling in perhaps opposite directions, but not always where one cancels out the other, but they can be working collinearly and therefore generate a new homeostasis, sometimes a dysregulated, dysfunctional, pathobiological homeostasis when they're dysfunctional together. So what else we want to say here? All those sphingolipids we just mentioned there, 2 major families, of course, participate or are necessary for healthy central nervous system molecular physiological phenomena. They include neurogenesis, repair, and event ontological learning, that is learning, knowledge-based learning, through environmentally ascended epigenetic reprogramming of gene expression. Certain sphingolipids are required for programmed cell death, like ceramide, and the suppression of that terminal pathway. So both things can occur, as I said. So they could be contrary, right? In human CNS disease, this is kind of an example here, like MS, right, multiple sclerosis, there's a multiple sclerosis associated oligodendrocyte degeneration. I think we all know that. Oligodendrocytes are non-neural CNS cells. And that degeneration has been demonstrated to remit and relapse according to synthesis and turnover of key competing and I would say maybe compromising sphingolipids. I like that word, compromising sphingolipids. So this asterisk is for me, reminding me to tell you a little bit about cyclosine right now. I'm going to get into it, in fact, right after this slide, a little bit more about cyclosine. Really an interesting galactosyl sphingolipid. So galactosyl sphingosine or cyclosine is potentially toxic glycosphingolipid. Glyco just simply means it's covalently bound to carbohydrate, in this instance, galactose. And in fact, it accumulates in a very terrible demyelinating disorder called the Krabbe disease. Okay, so that particular sphingolipid accumulates. Now, there's a paper that I'm just going to introduce here, and we'll get into more detail later on as we get into these lectures. This paper just came out in PNAS, October of 2019, so just 3 months ago. And it reveals that the biosynthesis of sphingosine is the modulatory signal associated with that neuropathology and the accumulation which is associated with that neuropathology of this cyclosine galactosyl lipid. And so then it becomes a Diaeventontological parameter. That is the way or how you get to the synthesis of cyclosine is how you get the disease. It's not simply the accumulation. And that's why a Diaeventontological perspective of biological systems, in particular biomedicine, is so critical to understanding. We're not looking at substances, we're looking at events. That is, how do you get to the production, accumulation of specific compounds like the sphingosine? Okay, so take a look at that in a little bit more detail. So this is directly from that paper, and it's the role of the acid ceraminidase in ceramide degradation and cyclosine synthesis. So this first part here, part A, shows you this acid ceraminidase. Okay, so acid ceraminidase is going to take this ceramide. You notice it's an N-linked fatty acid here, see right there, and you're going to remove that fatty acid. Now you have just made sphingosine. See, so you've taken ceramide and made sphingosine. I already told you that these two act counter or contra to each other because they bind different receptors and do different things. So that's what acid ceraminidase does. So now you have free sphingosine. When you have free sphingosine, you can make then, see here's sphingosine plus galactose, and sphingosine plus galactose in the anabolic pathway, which involves both the Cleveland and Kennedy pathways, something that I will detail for you. Those are good hallmark, constitutive, canonical lipid biosynthetic pathways. You can make this galactosyl sphingosine or cyclosine. Now you can also take galactosyl ceramide, so that's what this compound is here, galactosyl ceramide, okay? And you can degrade it via the acid ceraminidase. You can remove that fatty acid directly, right? Because it's missing because you just have sphingosine. And the catabolic pathway, which was published way back in 1973 from those authors, will still give you the same compound. And what this paper is going to tell you is that cyclosine can be potentially synthesized either of the anabolic dehydration pathway of sphingosine and galactose or through the catabolic deacylation of the preformed galactosyl ceramide, right? And they're going to tell you that acid ceraminidase, right, that enzyme there is going to cleave that amide bond to that fatty acid there. That's going to be the major pathway for making cyclosine that's involved in the Krabbe disease. So we're not making it anabolically, we're making it catabolically. And why does that matter if it's the same compound? Because the regulation of catabolism is unique and differentiated and indeed non-disinterested. That is, it is very specific and particular about the accumulation of that cyclosine and then the ultimate pathology of the disease. That's because the regulation of the production of cytosine is going to be totally different than the anabolic pathways. Okay, so that's what I mean by a dialectical event ontological perspective. We need to know how things are made, how you get to produce a substance. So it's not about the substance, it's about the event that allows for the accumulation, turnover, utilization, movement, trafficking of particular compounds, some of which can be directly cytotoxic, like this compound cytosine. Okay, so now you get it. All right. Now, let's bring it to now another focus. Now, what I do, if you've never watched me before, is I take multiple papers that have been published in the primary peer-reviewed scientific literature, right? And what I do is I say, okay, I want to know the entire focus of a given scientific discussion. So I can't just read one paper and get everything I need to know. I need to be able to grasp an understanding of multiple papers to be able to come up with a synopsis of what's going on in the biomedical pathway that I am trying to confer knowledge to for my clients. And what I'm doing here with these lectures is showing you how it's done and plus giving you some new information. So this is just a ritualistic pro forma video lecture so that you get to see what I do. I'm actually teaching you something because what I'm doing is I'm introducing to you an entire subject, right? How autoimmunity can lead to neuropsychiatric disease. I'm going to show you with a florid understanding of all the pathways, because I'm a biochemist, and then utilizing all the papers that we're discussing, a full description of how the system can possibly be functioning when things go dysregulated and dysfunctional in the pathological disorder. So here's a paper published in Neuromolecular Medicine a couple of years ago, March 2017, almost 3 years ago now, 2 years and 10 months ago. So we know that multiple sclerosis is a demyelinating neurodegeneration of axons accompanied with a massive demyelination overall. Now there are microRNA, and the microRNA expression is associated with other epigenetic modifications, including DNA methylation, particularly cytosine residues, and histone acetylation, particularly lysine residues with H3, histone 3, but not necessarily only that one. Those play clear roles in multiple sclerosis severity and progression. That whole idea of remission and relapse, right? So we're talking about a retailoring of the myelination of those axons, and then the constant remission, relapsing of that disease, ultimately leading, of course, to complete neurodegeneration and death because of the demyelination. So that has to do all those phenomena there, that microRNA, acetylation, methylation, those are all epigenetic phenomena that control and differentiate gene expression. And that's post-genetic blueprint of what you have in any given cell, because that's an alteration of gene expression. So the subtle alterations in the regulation of epigenetic machinery, those three microRNA, acetylation, methylation, are expected, but severe shifts in methylation, acetylation, and microRNA expression can lead to a massive disequilibrium in gene expression, as I've been saying, centered on loci that are most necessary and therefore the ones most vulnerable to such modifications. Okay, so that's how you can target or focus on a locus of a specific genetic disorder, because the genes that are involved in that genetic disorder are the ones that are in most transcriptional or post-transcriptional translational play. And because of that, modifications or permutations in epigenetic reprogramming can be then focused on the dysregulation, because those are the genes that are being expressed specifically, coherently associated with a given neurological disorder. So multiple developmental and indeed senescent aging processes, cues, are made plastic and or elastic by disease-causing agents. Okay? And that could be, for example, bacterial, viral disorders, as well as stress phenomena, as well as anything that happens endo in terms of dysregulation of gene expression or lipid modification or membrane turnover. That's why senescence can play a major role. The older you get, the more fault you get in recombination repair of DNA, and that can lead to epigenetic changes that ultimately can lead to inappropriate gene expression or mutations in genes, and then the inappropriate expression of those genes because of epigenetic modification, sometimes to partially preventive repair, such as with PARP or with regular recombination repair, can lead then to a fixed mutation because of epigenetic reprogramming. Okay? And that's what I mean by plastic or elastic. Plastic, you leave an impression. Elastic, you have an event ontology, and then it's fixed, or that is, you don't know it ever occurred because the initial induration, right, the initial problem with the inveighing issue on that expression of that gene is no longer there. So the how you got there now becomes erased. But now you're dealing with an event ontology, which is entirely different, unique, and then you have to retrace and find out how you got there. And that was going back to that cytosine production I just talked to you a minute ago from that brand new paper. All right. So in MS and other neuroimmune diseases, specific genetic loci may be modified epigenetically, that methylation, acetylation, microRNA, major canonical mechanisms, and thus create a dysequipoise, right? So you have equipoise where everything is homeostatic and the controlling mechanisms are giving you a healthy cellular environment. So dysequipoise, where you disrupt the equipoise of all the expression of genes and turnover of membrane lipids, for example, you get that of heritable methylomes and acetylomes, which again are epigenetic phenomena, but you can inherit them. You can. And they manifest as disease remission, relapse, and even familial risk associations. See? They might look genetic, but they're actually epigenetic. In origin, that is how you get there. All right. Now, back up a little bit, because many of you don't know much about epigenetics. So I want to make sure you get an idea of what we mean by it. There's no sense in me teaching you something very sophisticated and brand new from the scientific literature if you don't have the foundational and coherent background, so that you know small truths, truths with small t, so that you get a better clearance of the scientific phenomenon, so that then I can introduce new scientific phenomena to you, and I can then make a coherent understanding of what the current biological, biomedical literature is saying of a given disease state, so that I can help out my clients. So the human phenotype is influenced directly or in combination with our genes and the environment. There's a genetic predisposition to many diseases, indeed, but our environmental stimuli involved mechanistically with phenotype. Indeed, they are. Epigenetics is the study of changes in gene expression independent of genotype. Independent. Doesn't mean genotype can't play a role, but the epigenetic phenomena is independent of the genotype, sensu stricto. Okay? You have to understand a little about modality in terms of phenomenology to get that idea, right? Independent doesn't mean there isn't a genetic basis. It just means the epigenetic phenomenon in the disease itself is not dependent on a given programmed genome, right? Doesn't mean there isn't one there. Okay. So, independent of genotype, that in certain instances are transgenerationally heritable. Okay, so you have to understand you can inherit epigenetic reprogramming. The epigenome then consists of a canon of DNA and histone modifications that influence gene expression by inducing conformational changes, biophysical changes, including chromatin remodeling. This then helps specify tissue-specific and temporal gene expression in normal healthy tissue as well. So, aberrant epigenetic profiles are associated with numerous diseases. We know this from the literature. Environmental stimuli can induce epigenetic modifications that, in turn, modulate gene expression. Furthermore, microRNAs impact protein abundance directly because microRNAs work at the level of basically erasing the level of messenger RNA of specific genes because of complementarity, homologous complementarity of the microRNA to the RNA that's being targeted, right? Sequence homology. Impact protein abundance through post-transcriptional, right? Because you have to have the messenger RNA already, that's post-transcriptional, regulation of the mRNA, and the expression of numerous microRNAs become dysfunctional with disease. So it's not only that you have this microRNA made, you have numerous types of microRNA can then cause dysfunctional expression of multiple genes. It's an epigenetic phenomenon. Physical exercise is an environmental stimulus that impacts health and well-being through life. We know this. It's also a potential trigger for epigenomic remodeling. This is exactly how tissue that is muscle tissue remodeling can occur because of exercise. Of course, it can also lead to excessive reactive oxygen buildup, production in the muscle, which can lead then to degradation of muscle tissue, depending on the source of the biofuel, fatty acid versus carbohydrate, and then the movement of fatty acids and carbohydrate through circulation and then modifying expression of transport mechanisms, either fatty acid transport mechanisms like CD36 or, of course, glucose transporters into muscle cells, the accumulation of intramuscular triacylglycerol, utilization of that for long-term muscle endurance versus quick twitch phenomenon associated with glycolysis and anaerobic fermentation of glucose to pyruvic acid and the production of ATP that way, right? For example, the health and fitness benefits conferred by regular aerobic and resistance exercise training include an alleviated risk of developing and reduced severity of various diseases: cardiovascular, metabolic, pulmonary, hepatic, renal, and indeed direct immune diseases. Obesity and cancer are other obvious pathologies impacted both phenotypically and epigenetically by exercise. And so this is where you have the problem in aging then superimposing its effect on the lack of repair of dysfunction. Exercise does impact the signatures of the epigenome, including DNA methylation, histone acetylation, and microRNA expression and turnover. And those molecular events can alter gene expression and ultimately phenotype. And why am I bringing up exercise here? Because indeed, talking about the human body, nutrition and exercise play a major confounding, competitive, and indeed neurovascular role in whether or not disease progresses or even incident in a given human. And all of those are dialectical eventontological perspectives at the individual level, at the individual agency level. Individual agency meaning, do you exercise and what's your diet like, right? Because you are the agent that decides on how much exercise you get and what kind of diet you have, especially how you age, right? It's really important to understand there's a dynamic system. So that's what this is trying to show you. Phenotype can have environmental influences, and you determine a phenotype by inheritance and genetics. This is the given genetics from mother and father, right? And then you inherit because of recombination. It's not a stochastic inheritance, but it also isn't a 50-50 inheritance, right? The two are different. If you don't know stochastic, look it up. It's more like this, where you have environmental influences, non-inherited epigenetic phenomena, genetic imprinting, which can be epigenetic phenomena, and you inherit genetics directly, like here, and you also get an inherited epigenetic recombinational effect or determination that can impose effects either directly. Environment can cause that, but then that can induce changes in inheritance, and then that can lead to a determinant phenotype. So this is more what's going on in the biological system versus the old-fashioned simplified idea that genetics, whatever the inheritance is because of homologous recombination and homologous end joining of specific genes and the way that recombination plays out after embryogenesis or during embryogenesis, right? After gametic syngamy, that all does play a role. But think now about how all of that is influenced by the environment. The environment alters gene expression. That can be inherited, and that can then play a role on genomic imprinting. And all of that can ultimately determine not only a phenotype set in space and time, but one that is elastic and plastic through space and time. Yeah. All right. So what are some key terms here? Chromatin. Chromatin is the elongated condensed form of DNA housed inside the cell nucleus in eukaryotes. DNA methylation. It's an epigenetic modification encompassing the addition of a methyl group to a DNA CpG island and indeed non-CpG sites, but that methylation is going to be on the carbon 5 of the cytosine residue in the DNA. The enzyme itself that's carrying out the methylation, DNA methyltransferase, actively performs and catalyzes DNA methylation. and the methyltransferase itself. The enzymes are multiple, and they have specific specificity, and they turn over in the cell. The epigenome, what do we mean by that? All epigenetic modifications, including DNA methylation and numerous histone modifications, methylation and acetylation being two forms, described elsewhere, which I'm going to describe. Those epigenetic modifications all share common characteristics, however. Specifically, they act on DNA or proteins associated with DNA packaging within the nucleus. And they manipulate the structure then of the actual condensed chromatin. A biophysical change in condensed chromatin, going from euchromatin to heterochromatin, and alterations thereof are biophysical phenomenon that are translated through biochemical paradigms and ultimately alter gene expression. So in doing so, this epigenomic mechanisms either encourage or repress transcription, depending on the location of the modification of the genome, right? All right. Now, histone acetylation, chemical reaction involving addition of acetyl molecule, acetate to histone proteins, usually lysine residues, almost always, I think canonically. Histoacetyl transferase, you have an enzyme to do it. Family of enzymes called HATs, like that, HAT, and they catalyze histosylation. Of course, this enzyme, and indeed that methyltransferase, you have to make it to the nucleus. There's a whole other control over how those proteins are translocated into the nucleus and whether or not they turn over within the nucleus, whether or not they can be active, and they can be modified when they're in the nucleus too. Sequester, for example. Then there's a histone deacetylase. These are really important enzymes. They pull off acetate, right? They remove acetate via hydrolytic reaction off of histone proteins. And some of those histone deacetylases are called sirtuins. And sirtuins we've covered in multiple VerevMed and authentic biochemistry lectures. We'll go back and we'll talk about that again. Every time I do, I will reintroduce what I mean by it. But some of those enzymes are called sirtuins, those deacetylases, right? Histone proteins, of course, is constitutive of 5 in the eukaryotic system. Highly homologous histone proteins, there they are. They're called the H proteins, right? H1, 2A, B, 3, and 4. They're involved in DNA packaging, so you make chromatin. 4 paired histone proteins like A, B, and H3 and H4 form what's called an octamer, right? You get 4 pairs, you get 8. And they contribute to the formation of the nucleosome, right? And then that allows the packaging, but also controls gene expression. And then you have to have topoisomerases and gyrases to open up the condensed chromatin so that you get gene expression as transcription from the DNA via the leading strand, for example. Lysine, of course, is just amino acids found in proteins, and particularly in lysine residues and histones, look at acetylated. And then you have microRNA, small non-coding RNA molecules, approximately only between 18 and 24 bases in length. Remember, these are not DNA, these are RNA. Then the nucleosome itself is a repeated condensed structural unit of DNA packaging consisting of those octet of histone proteins wound up with somewhere around 250 nucleotides. So it just shows you have chromatin remodeling that goes on. That's how you get gene expression. So you get methylation patterns on chromatin, you get methylation patterns, acetylation patterns on histones. They are going to be modified in such a way that they are going to interact with the DNA methylation. So you have histone modification, DNA methylation alterations. All of that is going to change, that's up or down, the transcription of DNA off that template that has either that methylation, acetylation, very specific, cogent, coherent modifications associated with epigenomic reprogramming, aging, for example, on the nucleosome itself. And again, you get decreased or increased abundance of those methyl groups, acetyl groups, and then all that ultimately is going to lead to changes in RNA transcription. And of course, regular RNA transcription is going to be translated to protein. But then you have microRNAs, which are also synthesized, which are going to bind to the messenger RNA. And in almost all circumstances, when microRNA is targeting messenger RNA, it's going to decrease the expression of that gene. So a little bit more about microRNAs. They're short nucleotide, 20 to 24 RNA, ribonucleic acid, non-coding RNAs that are involved in post-transcriptional regulation of gene expression in all multicellular organisms so far examined. And they affect both stability and the translation of the mRNA. So they're therefore gene expression. MicroRNAs are transcribed by RNA polymerase II as part of a capped and polyadenylated primary transcript so that you get microRNAs or pre-pro microRNAs. They can be either protein coding or non-coding. Sometimes they can be, but they don't code, but they could be, you see. Difference in modality again. Necessary versus what is. Right? Primary transcript is cleaved by the drosha-ribonuclease 3 to produce an approximately 7 nucleotide stem loop precursor called the microRNA, the pre-microRNA, further cleaved by a cytoplasmic dicer ribonuclease to generate the mature microRNA and antisense microRNA star products, which ultimately then lead to the ablation or decreasing expression of the targeted messenger RNA. The mature microRNA is incorporated into RNA-inducing silencing complex called the RISC complex. That recognizes target messenger RNAs through imperfect, not absolute necessary, that it is completely homologous, base pairing with the microRNA, and most commonly results in translational inhibition or at least destabilization of the target messenger RNA. All right. This puts this all together. You get the pre-microRNA made. Pre-microRNA goes to this dicer complex. You have multiple proteins that are involved in making the RISC complex. That's then going to cause post-transcriptional repression of gene expression. Here you have the entire methylation, acetylation program, although this is, again, more epigenetic phenomena. Epigenetic modifiers, the DNA methyltransferases, for example, the demethylases, the HDAXs, or the deacetylases. And you're going to have all this different alteration of gene expression, okay? And all that alteration is going to either adenylate, destabilize, you're going to get translational repression, or you're going to actually get sensu stricto messenger RNA cleavage, all of which is going to lead to the repression of a specific messenger RNA expression. This is just putting it all together. So epigenetics, the microRNA regulatory circuit, and that's what we're looking at here. I'm not going to go through it again. Here are some well-described microRNAs. You can go into literature and you can find epigenetic regulated. These are themselves epigenetically regulated. That is the production of the microRNA. So a discussion now, let's back out about the Diaeventontological record. NeuroImmune pathways mediate central nervous system development and maintenance. Epigenetic mediated gene expression in neurons and glia that network within the CNS establish neural tracts and transmission in the healthy CNS. An autoimmune inflammation, of course, is a major disease paradigm in the CNS. Again, think about demyelination diseases like multiple sclerosis. And they may be linked to degeneration and neuropsychiatric sequelae. And that is the focus of all of these lectures, this one and all the ones I'm going to be presenting to you for the next several weeks. All right. Now, back to this paper published in Cell Death and Differentiation in 2019. There is the full complete citation. First author is Fogarty. Both neural stem cells and progenitor cells, these are neural precursor cells called NPCs, generate respectively differentiated neurons and macroglia. Okay? In the mouse model, these NPC pools expand from embryonic day 9 to 10, and neurogenesis begins at about embryonic stage 11 when the NPC exit the cell cycle and initiate, they're in the cell cycle, then they start initiating differentiation with programmed cell death, specified PCD apoptosis mediated, controlling the total number of cells in the nervous system. That's how the mirroring system functions to control that total neuronal load within the CNS. Now, a B-cell lymphoma 2, BCL2, pro-apoptotic plus anti-apoptotic proteins officiate that apoptotic program. You need the apoptotic program to control the amount of NPCs, right? You don't want to have an overproduction of these neural precursor cells because you want to differentiate what you've got, because otherwise you're not going to have any function, right? Think like fibrocytes in, say, muscle tissue after damage. They're not myocytes, right? So you don't want to have a lot of NPCs. You want to differentiate them into what? Into glia and all the various kinds of glia subclasses, and into neurons and all the different subclasses of those neurons, right? Otherwise, you don't have a differentiated CNS. It doesn't function, right? So you got to do a lot of apoptosis. You have proteins that control those. Are B-cell lymphoma 2. They were first described in B-cell lymphomas, okay? but these proteins are normal pro-apoptotic, but the family actually has anti-apoptotic proteins. So there is this seesaw up and down, right? Ride my seesaw type of ratio, where you have some pro-apoptotic BCL2 proteins. It's just called BCL2 because they were first described in B-cell lymphoma. It doesn't mean you have lymphoma when these proteins are expressed. Full stop. Don't think of them that way. Just the way they're named. And the second thing is you have same family of proteins that can work contrary to the ones that are pro-apoptotic. See, that's the whole point. In particular, it is the anti-apoptotic members, the myeloid cell leukemia, this is now a subclass, MCL1, and the BCL2 related gene long isoform, BCLXL. So these are subclasses of this BCL2 family, okay, appear indispensable for mammalian nervous system development. It's not just murine, but also human, of course. Functioning hierarchically, so that means through space and time, so it's an event ontology, through neural development. But what are the details, and how is the program hijacked, for example, during tumorigenesis? So we're going to discuss MCI and BCLX, but we're not going to do that now. We're going to stop here, right? This is going to just be the first lecture to give you kind of the taste of where we're going with this. And what I want to do ultimately is get you to where you can use this first set of lectures, right, the ones we just discussed here, and get us back to this whole larger picture of where I'm going to take you with the papers we're going to examine, which are going to involve this entire neuropsychiatric association that is linked to autoinflammation. In the CNS, primarily, I'm going to focus on those papers, but you're going to find out that you can get inflammation in the periphery, even in non-neural tissues, and they can still sense and target to the CNS and induce neuroinflammation in the CNS. And that inflammatory response can be linked, yes, to neurodegeneration, to demyelination, and also from papers we're going to be discussing subsequent to this lecture, a whole host perhaps of neuropsychiatric disorders, some of which are, again, are very much associated with autoimmune phenomena. Okay. All right. So I'm going to stop here, and I'm going to say this is Dr. Daniel Guerra saying bye for now, and we'll see you soon.